Welcome to the class. Today's topic is plant transformation method, and this is the part 2 of this lecture. We were discussing different vectors for plant transformation needed, and we had discussed co-integrate vector. Next vector is binary vector. This binary vector is a pair of plasmid. In this pair, one is known as helper plasmid and the other is known as disarmed Ti plasmid. Disarmed Ti plasmid, that means the oncogene which causes crown gall disease in plants, has been removed. And these genes are responsible for synthesizing various hormones in the required for crown gall disease in the plants. This is having a left border, right border. These are multiple cloning sites. This is plant transformation marker gene. And this helper plasmid is having only virulence region, no other region. Required for both cases for E. coli and agrobacterium. There are left borders and right borders that limit the tDNA region. A plant transformation markers, that is PTM, for example, neomycin phosphotransferase 2 and PT2, conferring kanamycin resistance is present here. Multiple cloning sites for introducing our foreign gene. A bacterial resistance marker, for example, tetracycline resistance gene for selecting binary vector colonies in E. coli and agrobacterium. Oride T sequence for conjugal mobilization of the binary vector from E. coli to agrobacterium is there. And there is a broad post-range origin of replication such as RK2 that allows the replication of binary vector in agrobacterium. Production and use of binary vector. The foreign gene of interest is inserted into the multiple cloning site of the binary vector in the disarmed Ti plasmid. In this way, the target gene is placed between the right and left border that repeats and cloned in the E. coli. So, this disarmed Ti plasmid is cloned in E. coli, whereas the helper plasmid is replicated only in agrobacterium, as it has origin of replication for agrobacterium only. And then by a mating process, the binary vector is mobilized from E. coli to agrobacterium, where the virulence gene of helper plasmid facilitates eDNA transfer into the plant cells. What are the advantages of binary vector? It involves only the transfer of a binary plasmid to agrobacterium without any integration. This is in contrast to co-integrate vector system. And binary vectors are more frequently used than co-integrate vectors. Plant transformation technique. How agrobacterium mediated transformation is operated in the lab. Agrobacterium mediated technique is the most widely used for the transformation of plants and generation of transgenic plants. Important requirements for gene transfer in higher plants through agrobacterium mediated transformation are follows. First, the explant. Explants are the plant parts which are taken for gene transfer here. They must produce phenolic compounds, for example, acetylsteronzoane for activation of virulence gene. If such compounds are not produced by the plant cell, then these compounds can be added into the medium also. Transformed cells or tissues should be capable to regenerate. So there should be a regeneration protocol in advance that should be optimized in your lab. In general, most of the agrobacterium mediated plant transformation have the following basic protocol. We will take our explant, that may be immature embryo or embryonic alive. Then pre-culture treatment is given. After that, liquid co-culture is done. Liquid co-culture, that is the suspension of bacterial culture in which your explants are suspended. After this treatment for 10 to 15 minutes, co-cultivation is done. Co-cultivation is transfer of these explants treated with bacterial culture onto the solid medium. They are allowed to grow for around 2 to 3 days, that is known as intermittently. For this, a bacteriostatic medium is prepared, that means medium having one antibiotic. Once a plant is growing and the agrobacterium growth is stopped, then the selection will be carried out. Selection is carried out with the help of the antibiotic for which antibiotic resistance gene is present in the gene construct in the plasmid. After selection, there will be viability of only transformed cells in this medium, and then these explants or these cells are regenerated into the plantlets. So there will be shoot induction, root induction, and it will lead to a establishment of a healthy plant that is known as transgenic plant. So this is the basic protocol for raising transgenic plants via agrobacterium mediated transformation methods. So first there is development of agrobacterium, which is carrying the cointegrate or binary vector with the desired gene. This agrobacterium is grown in a liquid medium. Then identification of a suitable explant, for example, cells, protoplast, tissues, callus, and organs. Then co-culture of explants with agrobacterium. Then killing of agrobacterium with a suitable antibiotic without harming the plant tissue with the help of a bacteriostatic medium. Then selection, and last procedure is regeneration. Advantage of agrobacterium mediated transformation. This is a natural method of gene transfer. So only the natural method which is present in the natural condition is being manipulated here. Agrobacterium can conveniently infect any explant, maybe cells, tissues or organs. Even large segments of DNA can be efficiently transferred. Stability of transferred DNA is reasonably good, and transformed plants can be regenerated effectively. Limitations. There is a limitation of host plants for agrobacterium since many crops, monocots like cereals, are not infected by it naturally because these plants are not producing acetosyringone. So, in such cases, acetosyringone has to be added synthetically into the medium in which agrobacterium mediated transformation is being carried out. The cells that regenerate more efficiently are often difficult to transform. TI plasmid that carries out this transformation contains one or more tDNA region that is integrated into the plant host genome to size 12 to 24 kb. Right and left border sequence which will be transferred into the host plant genome containing tDNA sequence, and it contains a Vir region. Vir region is entire box of different Vir proteins. It is having at least 8 to 11 Vir genes which are required for the transfer of tDNA from agrobacterium to the plant cell, and its size is around 40 kb. Process of tDNA transfer and integration. So first step is signal recognition by agrobacterium. The agrobacterium perceives signals. From where the signal is coming? The signal is coming from the sugar and phenolic compounds like acetylsyringone, which are released from plants when got wounded in natural conditions. Your plants will get wounds. They will start secreting of some substances like sugars and acetylsyringone. Agrobacterium will receive such signals and then it will be ready to enter the plant cells. Then second step is attachment to cells of the plant. So initial attachment happens via polysaccharides. And bacteria produce cellulose fiber mesh, where genes involved in the attachment of bacterial cells to plant cells, and this attachment is stable due to the signal perception of acetylsalicylate and sucrose. The weird genes are induced. So there is induction of weird genes. First weird gene that is induced is Vir-A. Vir-A senses the phenolic and phosphorylating due to activated Vir-G gene. Vir-gene's expressions are induced by Vir-G. So that is the function of this Vir-G gene. It induces all other Vir-genes for their function. T-strand production is the next step. So the Vir-D1 that is bound to right border after binding, it relaxes supercoiling that induces the action of Vir-D2. Vir-D2 has endonuclease activity and it binds to the 5' end. Similarly, at the site of nick, 3' end produced and serve as a primer. As a result, Vir-E is displaced by a single strand of tDNA. The tDNA is again nicked at left border to generate a single-stranded tDNA copy. The protection of Vir-E2 is done by nucleases. Vir-B is necessary for virulence, which has ATPase activity, and it also helps in tDNA delivery into the plant cell through a nuclear pore complex. Transfer of Vir proteins and tDNA into the plant nuclear localization. The single-stranded tDNA is converted to double strands by replication in the nucleus, and this double-stranded tDNA integrates from multiple sites into the host genome by unauthorized recombination. Practical application of agrobacterium mediated plant transformation is considering to induce less rearrangement of the transgene, and it is very successful in various plants. So these are different Vir genes and these are the function of these Vir genes. First are Vir-A and Vir-G. These senses phenolic compounds from wounded plant cells and induce expression of other virulence genes. Vir-D2, it has endonuclease function and it cuts tDNA at right border to initiate T-strand synthesis. Vir-D1 is having topoisomerase activity. It helps Vir-D2 to recognize and cleave within the 25 base pair border sequence. Vir-D2 also covalently attached to the 5' end of the T strand, thus forming the tDNA complex and also guides the tDNA complex through the nuclear pores. Vir-C binds to the overdrive region to promote high efficiency T strand synthesis. Vir-E2 binds to T strand, protecting it from nuclease attack, and intercalates with lipids to form channels in the plant membrane through which the T complex passes. Vir-E1 acts as a chaperone which stabilizes Vir-E2 in the agrobacterium. Vir-B and Vir-D4 assemble into a secretion system which spans the inner and outer bacterial membrane, required to export of the T complex and Vir-E2 into the plant cell. mediated transformation in plants where agrobacterium is transferring the cDNA into the plant cell, and this transformed plant cell is being regenerated into the plant with the help of tissue culture methods. So these are the references which you can follow. First paper is agrobacterium mediated plant transformation, biology and applications. The link has been given here, so you can download these papers also. Otherwise, you can Google, you will find them as such. Second paper is agrobacterium mediated transformation of tree food crops, methods, progress, and challenges. That is all about the plant transformation method via agrobacterium mediated transformation. That's all from my side. Thank you so much for listening carefully.